Namaste students, I am JVN Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Vegetable Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic removal of intracellular products from the downstream processing. So what are the techniques in separation of the intracellular product separation? A few product recovery methods may be considered to combine two or more stages. For example, expanded bed adsorption accomplishes removal of the insupers and product isolation in a single step. Affinity chromatography often isolates and purifies in a single step. Okay, so these are the methods which are used. The processes of the downstream processing include some techniques, which include the first step is the cell separation. The cells are separated by the flocculation, centrifugation, and the filtration. So there are different materials are present in the when the whole fermentation process has been completed. There are different products which are present in the um, uh, fermentation methods, which include some cells, intracellular products, extracellular materials, enzymes, proteins, okay, antibiotics. So for that, those separation, the first step is the separation of the separation. Cell separation is conducted by the flocculation, centrifugation, and the filtration methods by which we can separate the cells. Then cell disruption can be conducted for the isolation of intracellular products. Okay. For isolation of intracellular products, some techniques such as the homogenization and hydrolytic enzymes are used uh, for the for the destruction of the destruction of the cells by mechanical, physical, and the chemical methods. And after that, we can also classify the products by the centrifugation and the filtration methods. Then we can also complete this product purification by the concentration, and the techniques of the concentration include the precipitation, chromatography, ultrafiltration, precipitation, and the distillation. Then we can go for the high resolution technique for the product purification, such as the chromatography, electrodialysis, and electrophoresis, dialysis. So these are the techniques by which we can purify our product efficiently. Then after this purification stop, the purification product should be processed for sending it to the market. And for that, we need to polish the product. And the polishing product include the crystallization method, filtration method, gel, chromatography, drying, sterilization. So these are the methods by which we can isolate a pure product. Okay, and in this lecture we are going to separate the cell from the medium. The recovery of cell and the medium is a clarification method. For intracellular enzymes, the cell fraction is required. For extracellular enzymes, the culture medium is required. If we want to separate the cellular enzymes, then we need to isolate the cell sections. So we need to go for the cell disruption, and so that we can isolate the proteins which are present in the cells, enzymes which are present in the cells. Okay, but we do not need any kind of cell disruption for the extracellular enzyme isolation. When we go for the industrial scale, the cell medium separation is always performed by the centrifugation. And industrial scale centrifuge may be batch centrifuge, continuous centrifuge, and continuous with a decelerating centrifuge. So first step is the separation of solid liquid separation. So how solid liquid are separated? Some steps are involved in the solid liquid separation. The first is the flotation. Flotation. What is the flotation? Gas is introduced inside the liquid. Okay, a gas is introduced inside the liquid, and when it is introduced, it forms some bubble. Cells and other solid particles are absorbed and they are removed. So what actually happens? This gas will flow to the liquid medium. It will make some bubbles. Okay, and it may be also possible that it is due to the respiration. Okay, if it happens, the bubbles happens, the bubbles are observed, then the cells and the solid particles are observed easily and they will be separated. The next is the flocculation. What is the flocculation? Flocculation is next step for the after flotation. Cells, we have already discussed about the flocculation. Cells and debris from large aggregates and that will settle down. An addition of flocculating agent will result in the floc formation. So what actually happens in the flocculation? We are adding some uh, charge. We are adding some charge to the products. The product will cause charging, and that results in the collide formation in the small particles. Okay, they will repel each other, and when we stop, we neutralize the charge, then the small material will collide each other and the larger particle will be separated and they will form some flakes. Okay, which is known as the flocculation. Next step is the filtration method. Filtration is most commonly used to separate biomass in culture medium, and we use some membrane filters for the separation. So what type of filtration I use? Some depth filter, they are composed of the filamentous matrix. Okay, the depth filters are composed of the filamentous matrix, and particles are trapped in the matrix and liquid are passed out. Some absolute filters are also used that are specific pore size than the particles to be removed. Mostly are used to remove bacteria. Okay, so these absolute filters are used to remove bacteria. What is the rotary vacuum filter? They consist of rotation drum immersed in the tank of the pot. The drum, when the drum rotates, it uh, picks up the mass and they will form a cake. So these rotary filter will rotate, okay, and when they will rotate, they will pick some mass of the of cells, which will make them into the cake formation. Okay, then membrane filters are these filters which are of different size. Okay, they can be used in different size membrane filters. And for separation of different molecules, different sizes of the membrane filters are used. For the separation of the small molecules, uh, membrane filter of the smaller pore size are used, and for separation of the large particles, membrane filters of the larger pore size are used. Next is the centrifugation method. What is the basic of the centrifugation method? if we are adding these materials inside a centrifuge, then the separation will be conducted on the basis of their gravitational forces. Or we can also say that the molecules, the part products are separated on the basis of their centrifugal force or their sedimentation. So which centrifuges are used? The first is tubular bowl centrifuge. Okay, tubular bowl centrifuges are small and are commonly used in the pilot plants. As the name indicates, tubular bowl. The samples is added in the tubes. Okay, these centrifuges are like some tubes. They have some tube-like structure, and they are commonly in the large particles, and they are run at high speed and can be run in both batch culture and the continuous fermentation. Next, the disc centrifuge. These centrifuges consist of several discs that separates bowl into the settling zones. Slurry is fed through the center. The next is the multi-chamber centrifuge. This is a modification of the tubular bowl type centrifuge, and they consist of several chambers which allow them in zigzag movement. Okay. These multi-chamber centrifuges are the modification of the tubular chambers. Okay, the tubes are in multiple chambers. Okay, in the tubular centrifuge, the tubes were in a single chamber, but in the multi-chamber centrifuge, the tubes are arranged in multiple chambers, and they consist of several chambers which allow zigzag movement of the feed. So this is all about the separation of the intracellular products from different methods. Here I discussed some methods such as the separation of particles by different methods, including the flocculation, flotation, filtration, and types of filtered, centrifugation. So these are the methods by which we can separate the intracellular protein, intracellular enzymes. So what kind of questions can be asked? The examiner can ask that explain intracellular product separation methods in the downstream processing, and which methods and which techniques are used in the separation of the intracellular product separation. They can also ask about the centrifuges which are used in the separation of the intracellular separation. They can also ask about the difference between the flotation, flocculation, and filtration. They can also ask about the solid liquid separation. They can also ask about the filtration, complete detail of the filter which are used. So my lecture is over here. If you have any question, you can ask me. Thank you very much.